Hello, everyone. My name is Brian Fernandez, and today I will be talking about trichloroethene. If you'd like, there's a chemical formula and structure to the right of the screen. So today we'll be talking about trichloroethene, and it is an environmental pollutant which leads to autoimmune diseases, and we're going to see if that's facilitated by the dysbiosis in the microbiome. So what is trichloroethene? Trichloroethene is an organic solvent and it's used for cleaning and degreasing in multiple places like manufacturing, cars, production lines for foods, and bottles. It is associated with autoimmune disease, so it can cause systematic lupus, erythematosus, and autoimmune hepatitis. There's little research on how trichloroethene affects the microbiome. And why is it important to study trichloroethene? Well, it is an environmental pollutant, so it is somehow going to be coming into contact with you or someone else. So exposure can be via inhalation or dermal contact, and this results in an immune dysregulation. And frequent exposure to this can be very detrimental, as we find later on. According to the Occupational Safety and Health Administration, around 3.5 million Americans are exposed to trichloroethene through means of inhalation or dermal contact. However, what is scarier is that 14 million people in the United States are exposed to trichloroethene through their drinking water, which is contaminated. And I want you to keep in mind this number that I'm about to say. It's OSHA's maximum limit of exposure, and it's 100 parts per billion, which is equivalent to 76 kilograms per milliliter per day. And this number will be shocking later on as we go through the results of the discussed paper. So, what is systematic lupus erythematosus? It is a chronic autoimmune disease where it affects many parts of the body and causes high levels of inflammation. Some conditions associated with systematic lupus erythematosus are pleural effusions, heart problems, lupus nephritis, arthritis, and Raynaud's phenomenon. What is autoimmune hepatitis? It's inflammation and targeted attacks on the liver, so not just systemic wide. This is more focused on the liver. And if not treated, it can lead to cirrhosis, biliary cholangitis, and sclerosing cholangitis. So the paper that we'll be looking at is written by Wang et al., and it is named Gut Microbiome Host Interactions and Driving Environmental Pollutant Trichloroethene Mediated Autoimmunity. So the researchers wanted to bring some insight into how the hazard of trichloroethene is an environmental pollutant and how it affects the autoimmune diseases and progressing with dysbiosis of the microbiome. And then once the researchers got a current understanding of the field and like how trichloroethene affects everyone and what is out there already, they came up with a hypothesis, and it's, does the microbiome contribute to trichloroethene mediated autoimmunity? And it's focused on systematic lupus erythematosus and autoimmune hepatitis. So the experimental design includes two populations of mice. So the first one is female mice MRL positive. So these are the ones that are treated with trichloroethene, and it's about 0.5 milligrams per milliliter a day. And it was used in their drinking water. And the total treatment was about for 52 weeks. And after that, they were euthanized. And for the next mice group, they were more of the control. So they got a baseline reading of what all their microbiome composition was, their lymphocyte count, their cytokine count, And after that, they were treated with broad spectrum antibiotic cocktails. So it mainly consisted of ampicillin, vancomycin, metronidazole, and neomycin. And that was also taken through their drinking water for 4 days. And then this is where they got the fecal matter transplant. And this was through PBS capsules. And then so the researchers quantified their experiment through microbiome analysis, and that was 16S RNA sequencing, and they used the V4 region. They looked at trends in taxa abundance, alpha diversity, and beta diversity, and if there was any bacterial changes, they used quantitative PCR. The next one was immune cell infiltration. So they did this by using flow cytometry to measure the amounts of CD8, CD4 T cells and where in the body, so systemic, and if they had any hepatic T cells. The next one they used was ELISAs and bioplaque assays. So this is where they use serum antibodies to determine mouse-specific antibodies for anti-nuclears, anti-double-stranded DNA, anti-smooth muscles. They also used MDA protein adducts in the liver and colon tissues. Those were analyzed, and they also used serum cytokines to determine which cytokines were present in the system of the mice or in the hepatic system. And quantitative reverse transcriptase PCR, that was used for taxa abundance when there was those changes, as I said, in the 16S RNA sequencing. And they also used Western blot analysis, and this was mainly to look at tight junction proteins, total colon tissues, and proteins of that such. And then they used statistical analysis to gather all that information into graphs so we can understand them very well. So this is where we're going to start talking about the results. Long-term exposure to trichloroethene induced an autoimmune response in the MR++ mice. So there was a significant increase in anti-nuclear antibodies and anti-smooth muscle antibodies. And since there was this low dose of 0.5 milligrams per liter per day, it was seen to exacerbate autoimmune disease responses. So there was overall systemic and inflammatory response. There was seen increased serum levels of interleukin 6 and interleukin 12. And the increase of anti-nuclear antibodies is consistent with systematic lupus erythematosus disease, and an increase in both anti-nuclear antibodies and anti-smooth muscular antibodies suggests an onset of autoimmune hepatitis disease. And so this graph here, we can visualize the results from the previous slide. This is figure 1A, and the anti-nuclear antibodies is shown in a much lower in the control mice than in the mice treated with trichloroethene, as you can see in figure 1A right here. And then in figure 1A and figure 1B, these align with the claims of how higher anti-nuclear antibodies is consistent with systematic lupus erythematosus, which is the condition where the whole system of the body of the antibodies target the whole host. And then in figure 1B, we can see that anti-smooth muscle antibodies are much lower in the control mice than in the mice treated with trichloroethene. And so when we take into account this whole picture, the increase of anti-smooth muscle antibodies in combination with the anti-nuclear antibodies is consistent with autoimmune hepatitis due to the anti-smooth muscle antibodies targeting the liver, as well as causing inflammation around the whole body, leading to more damage and this repeated cycle of this happening. And on figure 1C, you can see there is higher levels of interleukin 6 and interleukin 12. This leads to lymphocyte activation and proliferation. And this is not good when there's nothing to be attacking, no foreign antigen or pathogen is present. So the results for the splenic and haptic immune responses. So there's an accumulation of immune cells, and it's analyzed using through flow cytometry, as we said. And trichloroethane exposure in the spleen showed a significant increase in CD4 plus T cells. So those are activated helper T cells, and a reduction in macrophages. However, in trichloroethane exposed mice in the liver, CD4 plus T cells had no significant change. But however, they were producing more interferon gamma than the control mice in the liver. There was also a reduction in macrophages overall and dendritic cells in the liver. And PD-1 expression in both splenic and haptic CD4 T cells was also increased. And we'll see that in the next slide right here with the graphs. So this figure backs up the results stated on the previous slide, where we see how cells from the spleen and liver of trichloroethene treated mice with symptoms of systematic lupus erythematosus and autoimmune hepatitis ran through flow cytometry, showing the significant increase in CD4 plus T cells and reduction of macrophages in the spleen, as seen in figure 2a. However, the hepatic CD4 plus T cells did not see a significant change after trichloroethene, and the reduced percentage of macrophages and dendritic cells were observed and could be seen in figure 2D. So this cell activation that we see with CD4 T cells can also be associated with the programmed cell death protein 1, PD-1, an immune checkpoint that is mainly expressed by activated T cells and B cells, which infiltrate tissues and contribute to autoimmune disease progression. The decrease in tight junction proteins in combination with the autoreactive T and B cells expressing this PD-1 are what likely is the cause of autoimmune responses being higher within the intestinal gut and eventually lead to targeted organs like the liver. And this is where autoimmune hepatitis arises and systematic lupus erythematosus arise from. It's where that gut barrier dysfunction from tight junction proteins, which we'll talk about later on, and program death protein 1 being absent. So results of trichloroethene exposure alters the gut microbiome in the mice. This is where we actually see the microbiome being changed. There was actually no change in the alpha diversity, but there was beta diversity change. So there was bacterial community clustering and the types of bacteria present in the distribution levels. The changes were at different taxonomal levels, so 6 bacterial families and 8 bacterial genera. So in this graph, we see a reduction in bacterial families, Uraciopiolatricaceae and Lactobacilliaceae. Under Firmicutes phylum and bacterial families, Marinifilaceae and Reichionella. Under Bacteroidetes phylum in the trichloroethene treated mice as compared to the controls, as we see in figure 3D. This significant reduction in Bifidobacteriaceae and Uraciopiolatricaceae were observed in trichloroethene treated groups, as seen in figure 3D and figure 3E. And then at the lower taxonomal level, trichloroethene exposure led to significant decreases in the genre of Lactobacillus and Bifidobacterium, seen in figure 3D and 3E. These genera are also known as probiotics, and they're known to promote induction of T regulatory cells, and the reduced levels can thus promote these autoimmune diseases. And trichloroethene exposure, on the other hand, has also shown an enrichment and increase of Akkermansia and Osteobacter, and this was verified using the real-time PCR, and this is consistently tied to trichloroethane exposure and do significantly increasing the fecal Akkermansia monophilia, and this is seen in figure 3F. In addition, we also observed decreasing trend for Faecalibacterium protensi after trichloroethene exposure. And some other reductions at the genus level included Turibacter and Faecalibaculum under the Firmicutes phylum, and Alistipes and Orduribacter under the Bacteroides phylum. Some results were trichloroethene exposure led to intestinal oxidative stress and barrier dysfunction while regulating intestinal inflammation. So trichloroethene exposure led to this significant increase in MDA protein adducts. And these proteins are conjugated form of the lipid peroxidation product MDA with proteins and are also a measure of oxidative stress. The tight junction proteins measured were claudin 1, claudin 2, claudin 3, ZO1, ZO2, and oculidine. Colon protein levels of ZO2, oculidine, and claudin 3 were significantly decreased following the trichloroethene exposure. What was found was that trichloroethene induced microbiome dysbiosis, and this could influence overall redox system and gut integrity due to tight junction proteins being lower than that of the control mice. Figure 4D, E, and F have the purpose of showing if trichloroethene microbiome dysbiosis interacts with the host MRL++ mice, so the mice that were treated with trichloroethene, their intestinal immune system. And this is done by measuring CD14, tumor necrosis factor A, interleukin 1 beta, mucin 1, mucin 2, and lipocalin 2, known to be inflammatory markers. The mRNA level of colonic CD14, a transmembrane coreceptor for toll-like receptors, was significantly increased in the trichloroethene exposed mice. And this increase in interleukin 1 beta and reduction in mucin 1 at the mRNA levels, with lipocalin 2 and immunoglobulin A levels are all indicators of intestinal inflammation increasing. All this data therefore shows that trichloroethene, MRL++ mice, gut microbiomes were interacting heavily with intestinal and systematic immune system. So, fecal microbiota transplants from the trichloroethene treated mice generated an autoimmune response in the antibiotic treated MRL minus LPR mice, so the mice that were control mice, the ones treated with antibiotics. So the feces from the donor mice that were treated with trichloroethene and the control mice were both sequenced with 16S rRNA, and this was used to evaluate the microbiome composition and changes later on. So the alpha and beta diversity were significantly different before and after fecal microbiota transplantation. The family level had an increase in Bacteroideaceae, while a decreasing trend in Bifidobacteriaceae, Lactobacillusaceae, and Reikonelsaceae. And the oral feeding of the fecal microbiota transplantation through the capsules to the control mice resulted in a significant increase of serum levels of the anti-double-stranded DNA autoantibodies, so the ones that are attacking our own DNA, which is a disease marker of systematic lupus erythematosus. And anti-nuclear autoantibodies also had a similar trend. There was this significant increase of antibodies attacking anti-nuclear cells. What was also found interesting was that the hepatic lymphocytes and CD4 plus T cells had an increase in the control mice after the fecal microbiota transplantation. And this data is consistent with showing a causative role of the microbiome in trichloroethene-mediated autoimmunity. And some things I would like to discuss are there's increasing research showing how the microbiome can affect the whole system at large and be responsible for disease onset and be associated with pathogenesis of autoimmune diseases. So that low dosage of 0.5 milligrams per milliliter per day, that is very low dosage, and it was seen to cause a greater chance of developing autoimmune diseases. In the Occupational Safety Guidelines, so OSHA, their guidelines on trichloroethene exposure seem to be higher to not be an issue for workers. However, that wasn't seen in this research. It was shown that having a lower amount actually caused greater autoimmune disease onset. But what is even more shocking, besides the OSHA guidelines, is how 34 million Americans are exposed to trichloroethene through contaminated water. And we don't know how much is in this water. And it shows how our safety may be at risk due to the water we are drinking and the microbiota transplantation using the fecal matter from trichloroethene exposed mice increased hepatic inflammation, systematic autoantibodies in the recipient mice, and this further supported a casual role of the microbiome in the autoimmune disease. So just this little amount that we could be exposed to every day can be affecting our microbiome, and then that ultimately affecting our whole immune system, our whole body, causing these autoimmune diseases that attack our liver and every other organ and tissue in our body. And a limitation that I thought of throughout researching this whole article was how much the dosage markers weren't studied. They used one dosage marker. And I felt like, what if it was lower? What if it was higher? How would there be differences in the lymphocyte counts, cytokines, protein expression, and microbiome composition? Another question that I have for the researchers is, what was the diet of the mice like? They didn't really go into that. They just talked about the dosage through their waters, the trichloroethene for the experimental mice, and antibiotics for the control mice. And then just reading this whole article reminded me of hearing about forever chemicals and microplastics being found in newborns and no way of being safe from environmental pollutants nowadays. Researching trichloroethene affects the broader society because it brings to light how Americans and people around the world may be exposed to trichloroethene that can cause these chronic immune diseases and lower the quality of life for everyone. I can see how communities who may be struggling with access to clean water in the United States or in other countries could use research like this or research on other forever chemicals and how it affects our microbiomes and cause autoimmune diseases to bring change to the industrial usage of chemicals and how to make the environment safer for everyone now and for generations ahead of us. And who knows how long trichloroethene or other forever chemicals can stay in the environment or stay in our bodies and cause more damage. These are my references, and I'm going to leave everyone with some questions. The questions are, what do we think would have been found if there were multiple dosage types for the trichloroethene exposure? How can this research bring light to forever chemicals in the general population, so those who may not understand as much as me or you guys? And how should environmental pollutants be measured for safety? So that low dosage number, the 0.5 milligrams per liter per day versus the 76 kilograms per milliliter per day, how does that change? Because we did see how a lower dosage can still cause these great effects on the microbiome and autoimmune disease onset. Could a balance of probiotics with higher dosages of trichloroethene cause negligible differences in inflammatory markers and not cause autoimmune diseases? So like lactobacillus, like the good kind, can that help negate the effects of trichloroethene? How can we stop the problems of these forever chemicals getting in our systems and causing autoimmune diseases and changing composition of our microbiomes? And the last question is, what type of microbiome and immune response would we see if the experimental mice had a condition such as Crohn's disease? So how how worse will it be if these mice had a different disease or different symptoms with on top of their autoimmune diseases that were caused by trichloroethene? And with that, I will leave you guys to answer these questions and see you in the comments. Thank you.